So hello everyone. It's my pleasure to welcome everyone to right now already the third season of the originally we called it Optical Interest Group Seminar Series together with the MOIA Seminar Series at EMBL. But what we've right now did is we thought about over the summer to maybe rebrand us or rebrand the name of the series. That was also a suggestion by Christian Tischer. And right now, welcome to the Janelia EMBL Bioimaging Seminar Series, if that's not a great title. So we will use this title from now on to announce the seminar series to you and also the Janelians, and also when we later on distribute the talk, we will also use this branding. And the second new thing of our seminar series is that we not only have a talk with Q&A options, or with the option to ask questions in the middle, but also at the end. But what we will also have is we will, from now on, have a Meet the Speaker session. So that's a session which will be 30 minutes long, straight after the talk, straight after the presentation, where you actually have the option to ask more in-depth questions and maybe even get to know the speaker better. Why have we set this up? Why have we decided to go this way? Because we not only want that you kind of passively inhale what you get presented by the speaker, but that we also open up the option to for collaborations and even for networking between our both institutions. We will see how this works out. If that's a good way to do that, that's a good way to foster collaboration and networking. We will continue with the Meet the Speaker session. If it's not working, we might change the model also a little bit. So if you have questions, or if you have really in-depth questions about maybe also how to use Isabel's presented tools, maybe with your scientific research, you can stick in a little bit longer, and you can, and for that, please use the chat function to inform the host, either Christian or me, about your desire to get to know a little bit more. Otherwise, for short questions, for questions to clarify specific aspects in the talk, feel free to just interrupt in the middle or at the end of the talk for just very brief questions. So with that being said, let's welcome actually Isabel Espinosa Medina. So it's my pleasure to welcome you to the first talk of the third season of our bioimaging seminar series. And just to say a few words about Isabel. So Isabel is a postdoc in the Lee and Koyama labs here at Janelia, where she develops genetic tools for high-resolution lineage analysis in zebrafish and mouse. She earned her bachelor's in biology and biochemistry, followed by a master's in neurobiology and molecular biomedicine at the Autónoma University in Madrid in Spain. Afterwards, she moved to Paris to join the lab of Jean-François Brunet at the École Normale Supérieure to study the peripheral nervous system development. Here at Janelia, during her postdoc, she has implemented Tempo and TempoBo, new imaging-based genetic tools that allow differential labeling and manipulation of consecutive cell generations, revealing cell histories both in zebrafish and mouse. Today, she will talk about these genetic tools in her talk titled Temporal Encoding and Manipulation of vertebrate cell histories with a new CRISPR-Cas9 system. So the screen is yours, Isabel. Please take it away and share your screen first.
So thank you so much for the introduction and also for the invitation to talk at this seminar series. I'm very excited that I can share these new tools with both the colleagues at Janelia and EMBL. Again, as Ulrike said, I'm happy to answer questions briefly throughout the presentation or at the end, as you prefer. But especially if you don't understand something that is important for the continuation of the presentation, please interrupt me. So today I'm going to talk about new tools, imaging-based tools for temporal labeling and manipulation of vertebrate cell histories using CRISPR-Cas9. But first, I want to mention that my main interest in biology is to understand how it's possible that a single cell gives rise to the amount of complex tissues and organs that form complex organisms. Cells that organize in different patterns that interact with each other to make possible the functionality of a complex organism. Well, to answer to this question, scientists have borrowed a strategy similar to the reconstruction of family trees, actually, that is called lineage tracing. So lineage tracing means finding the relationships of every single cell of a tissue or organ between them and with the very first cells that originated them. But that alone is not enough. You also want the information on the morphology, on the function, and the molecular characteristics of the cell. But let's start by the lineage tree. That's already not an easy task, as you can imagine, because complex organisms are formed by hundreds to thousands or millions of cells. So tracking the lineage from the very first cell to the functional organism is almost impossible right now. But it has been solved for certain organisms. So the only fully reconstructed cell lineage is that of C. elegans that contains 959 cells and was solved by John Sulston, which actually earned the Nobel Prize for this and other research on the genome of C. elegans, who solves all the lineage tree of the C. elegans by looking through a stereoscope and recording by hand every single cell division. As you can imagine, this is not something you can do with other more complex organisms. You need other ways to track the cells, and actually new imaging technologies have emerged, especially, well, there's several of these research that went on here at Janelia to make this possible in more complex organisms, including vertebrates like zebrafish or mouse. And I'm showing you here two examples, efforts led by two amazing women in the lab of Philip Keller, Yinan Wan and Kate McDowell, who tracked the trajectories and the lineages of thousands of cells during the early development of zebrafish embryos or mouse embryos during gastrulation. This is incredible. What they achieved is also made possible by the amazing computational efforts and machine learning approaches that have been developed. But still, we cannot trace the lineages of cells past a specific time point in development. The limitation comes because the phototoxicity and the embryo development cannot be kept under a microscope for longer periods of time. Also, the cell segmentation in much more dense tissue structures makes the reconstruction almost impossible. So we also need other tools like genetic labeling strategies that allow us to trace the cells, segment the cells, or even reconstruct the lineages retrospectively. And that's, well, an example is that the complexity of the mouse brain, as you can see, contains millions of different types of cells, neurons, glia, endothelial cells. And really to reconstruct the lineages of this complex structure, you need something that allows you to do it retrospectively. You have a label that you can look at when the brain is fully developed and then reconstruct the lineages, inferring the relationships. Genetic tools classically that have been used include imaging-based or sequencing-based tools for lineage tracing. The classical tools based on imaging rely on the activation of fluorescent reporter on a progenitor cell normally by a recombinase that activates a fluorophore in a particular progenitor cell that then labels all the progeny from that progenitor with the same color. So that way we can know the lineage they belong to, but unfortunately we cannot know the temporal relationships between these cells. We cannot separate that these later born cells were born at a different time than the previous one because they are all labeled the same color. So although there's spatial resolution in these techniques, they lack temporal resolution. On the other hand, sequencing-based techniques allow us to reach an incredible resolution in terms of transcriptomic resolution, molecular characterization of each single cell, but you have to dissociate the tissue. So you lose the spatial information, the morphological information of the cells. and in many cases also the temporal information. So we need a tool that allows us both to have imaging and spatial information of the cells and temporal information. So for that, I have developed Tempo, that stands for Temporal Encoding and Manipulation in a Predefined Order. That is a genetic tool based on imaging that allow us to track different cell generations because we label them with different colors consecutively and in a specific predefined order, as you can see here. And it is based on CRISPR-Cas9 instead of recombinases because these provide us a more unlimited power to label things instead of recombinases that are limited in numbers. And is based also on a new type of genetic switches that we previously developed in the lab. Here with my colleague Jorge, we developed these CASA switches that are fluorescent reporters that are disrupted by a target for a guide RNA. They need that Cas9 and a specific guide RNA cut between two homology repeats of this fluorophore that is disrupted. And then by the repair mechanism single-strand annealing, that is a very conserved repair mechanism that every cell has, this fluorophore gets activated by a series of recombinations that eliminate one of the repeats and the intervening target sequence. So by CRISPR-Cas9 double-strand breaks and single-strand annealing, we created these switches that get activated. And how then I use this to develop Tempo? Well, I will show you. Here you have a construct that I designed that is expressing the fluorescent protein CFP. And the cells at the beginning will be expressing this reporter. Then, thanks to the CASA switches and CRISPR-Cas9 subsequent cuts and repairs, we get the activation of subsequent reporters in a predefined order. So after the CFP, we will get the repair of an RFP that at the moment it gets turned on, leads to the inactivation of CFP because it's in another frame. And finally, in the third temporal window of this cascade, we activate a YFP fluorescent protein, and then the RFP and CFP remain, in this case, inactive because they both are in different frames. Exactly how this happened in more detail, we call these ways of activating and inactivating fluorescent reporters frame switches. You have a sequence for a fluorescent protein in frame at the beginning, and you have a CASA switch that is inactive until CRISPR-Cas9 repairs it and place it in frame now, and the downstream reporter will be now inactive. Then how this cascade can happen in a precise order? Well, that is because we also implemented a cascade of guide RNAs. So similar to the cascade of reporters, we can actually trigger a cascade of guide RNAs thanks to the activation of Cas9, a guide RNA that is active, and a second guide RNA that is inactive at the beginning and then gets activated. How this happens? Well, this is what we call now a guide RNA switch. It's a guide RNA that is inactive because it has a target sequence for another guide RNA between its scaffold sequence. This scaffold, represented here as hairpins, is a structure of the guide RNA that is essential for its function. It needs to have this structure to be able to bind to Cas9. But when we disrupt it, it's not functional anymore. So the activation consists again of the cut by Cas9, the repair mechanism single-strand annealing that leads to its functional activation. So with these two components, we have a compact system. We just have two transgenes that allow us to activate a cascade of 3 colors in a predefined order. Each protein is expressed just once at a time. They are mutually exclusive frames. Only this single-strand annealing repair mechanism allow their activation. It's an irreversible system and occurs specifically in dividing cells. We can indeed activate the cascade in a tissue-specific manner if we lead the expression of Cas9 or the guide RNAs under specific promoters. If you have any question up to here, please don't hesitate. So we implemented this system first in zebrafish, validated how it worked by microinjection of one cell stage zebrafish embryos with the tempo constructs expressing these fluorescent proteins and Cas9 under the ubiquitous promoter in fish.
Huh?
And we, yes.
Yeah, sorry, I actually have a question, but maybe you say it. I did not understand when these switches happen. Is it just after 10 hours, or is it whenever a cell divides, or what?
You will see
okay.
this. Yeah, so this is part of the cascade of divisions during development. So depending on the lineage and on the organism, you have a specific timing. You have the option to activate it by yourself, but for that you will need some type of heat shock promoters that we have actually done before in Drosophila. So that's also an option. But for now, is happening on its own once you activate the cascade.
Okay, and then what? Okay, yeah, you will come to it, I guess. Yeah.
But yeah, no, feel free to.
No, but is it like then? So when is, for example, the switch from red to yellow? Like when the cell divides, or after some time?
So because it's, well, this is a process that depends on the efficiency of the cuts by Cas9. So it's under repair. So this repair mechanism happens during G2.
Okay.
So basically, that's where it has the option to happen. Doesn't mean that happens every cell division.
Okay.
It actually, from my data, it looks like it takes more around 2 to 3 divisions.
All right. Thank you. Mm-hmm.
So when we actually...
Yeah, so we have also another question in the chat by Cornelius Gross. He's asking, can you adjust the frequency of activation to control the fraction of cells marked and the timing?
So again, it will happen only if you control the amount of Cas9 or guide RNAs, and that can be done with specific promoters like heat shock promoter, for instance. So yes. Here I'm showing you ubiquitous continuous expression of the cascade just for validation. Any more questions?
I think we're good. Good now.
All right. So when we introduce these constructs in zebrafish, we first validated each independent frame switch transition to know that they are working as we expect. And for that, what you are seeing here is the reporter expressing CFP at the beginning. And in the absence of the guide RNA, we see, in this case, the zebrafish epithelial cells mainly labeled with CFP and not other color. But when we provide with Cas9 and the guide RNA that is specifically associated with the repair of RFP, we see that the majority of cells have turned RFP. More than 90% of these cells have turned on RFP, so it is very efficient and almost no leaking. We did the same for the other independent frame switch from RFP to YFP, and we also found even higher efficiency of repair of more than 90%. So this is a robust system, and the different frame switches work pretty consistently. For the guide RNA switch, well, this is the structure that I show you that we implemented. We had a first version of the gRNA switch that we implemented for Drosophila studies, and that had around a 50% efficiency of repair. This is the more detailed structure of a gRNA switch. Well, so in order to improve the efficiency of repair of this switch, which would be very interesting to increase the efficiency of the cascade overall, I designed a new gRNA switch in which I extended the hairpin region on both homology repeats, so that now the repeats almost double in length. This structure, even if the hairpin is longer, is still functional once the guide RNA is repaired, because it's similar to what bacteria used actually in the original guide RNAs. And after this implementation, we increased the efficiency of repair from 50 to 70% while maintaining the leakiness very low. So this is the assay that I used to study the efficiency of repair of the gRNA switch by using a reporter, in this case, that has RFP preactive and that needs the gRNA number 2 to repair YFP. But gRNA 2 is originally disrupted. So only in the presence of gRNA 1, we can see that there is repair and activation of YFP with the efficiency I just mentioned. So we then studied how this works in zebrafish, the full cascade, with microinjection of the ubiquitous tempo plasmids. We followed the progression of the cascade in developing embryos from 8 hours to 96 hours post-fertilization. And what we expect, obviously, is that we start with CFP, that transition to RFP, and then to YFP. And this is what we saw in this sequence. We first, at 8 hours of development, already saw CFP cells. In the nucleus, you have a white nuclear labeling that stands for Cas9. And subsequently, at 24 hours, we already see that most cells have turned on the RFP reporter. At 48 hours, the YFP reporter appears and then becomes predominant at 72 hours and continues to stay stable at 96 hours. Interestingly, those cells that cannot divide anymore, or they don't do in this time frame, like cells in A, labeled RFP, they don't transition to the next color as expected, while those that continue to divide, like the one shown in B, transitions to the next color, because single-strand annealing only happens in dividing cells. And here are the quantifications for this transition, showing that they happen in the predefined order, and that the latest color progressively accumulate. And the efficiency of color transition is high for both the first and the second steps. So now we wanted to see the functionality of tempo for other lineages, to see if we could reconstruct birth date for neuronal lineages in this case. And this was done in collaboration with Carla Belmonte from the lab of Cristina Pujades, that had been at Janelia, actually, had been at Janelia for a few months before COVID pandemic started. So we have continued to collaborate in the distance. So Carla and her lab described this ATO1A domain in the hindbrain of zebrafish that gives rise to important cerebellar structures. And it's a really good domain for proof of concept experiments, because they describe that there are two progenitor types that differ in their behavior. The ones in purple, the dorsal progenitors, divide only once before forming differentiated neurons and occupy the most lateral and dorsal parts of the hindbrain, while the ventral progenitors can divide 2 or more times and also stay in the most ventral and medial part of the hindbrain. So to see if tempo recapitulates these patterns, we injected ATO1A GAL4 zebrafish embryos that express the effector GAL4 under the promoter for this progenitor region, and injected them with conditionally activatable tempo constructs that will be only expressed in this case in the ATO1A progenitors. And what we saw is that tempo is activated in the expected sequence, also in this lineage. And in these orthogonal sections, we can see that there's actually a lateral medial distribution of the cells depending on how many times they divided. So you can see that those that divided less times, that express CFP, occupy more lateral positions, while those that occupy more medial regions are those that are YFP, so they divided more times. So as you can see, tempo recapitulates not only birth dates, because they follow the expected sequence, but also can help reconstruct past cellular migration and proliferation dynamics. Another example that I wanted to share with you is the assembly of locomotor VSX2 interneurons of the zebrafish hindbrain as well. In this case, these neurons are very important for locomotory behaviors and were studied in detail by Minoru Koyama in Janelia, where he recently published a study showing that birth date of these neurons is correlated with their function. Early-born V2A interneurons participate in crude behaviors such as escape responses, while late-born neurons are more important for more complex behaviors such as exploratory behaviors. So in order to see if tempo recapitulates this and also can expand the palette of colors that we can visualize, what we did was to generate, in this case, transgenic zebrafish lines that express tempo inducible by GAL4, in this case, in the interneurons of our interest, and another line that has Cas9 in neuronal progenitors. We cross these lines to specifically visualize tempo in the neurons of our interest. And here you see the results, a lateral and dorsal view of the hindbrain of zebrafish expressing tempo in these interneurons. And you can see that indeed the early-born neurons occupy more lateral positions as Minoru had described before. So here in the schematic, the purple cells will correspond to the CFP cells. While later-born neurons in RFP and YFP labeling occupy more medial positions. Then also orthogonal projections of the hindbrain show that, as was described before, the early-born neurons in more rostral portions of the hindbrain occupy more at the ventral region of this orthogonal section, while the latest-born neurons occupy more dorsal regions. On the other hand, in rhombomeres more caudal of the hindbrain of the zebrafish, it has been described that there's a migration where later-born neurons migrate towards more ventral positions later in development. So we also observe that in more caudal positions. And interestingly, when we look at the neuropil, the axonal projections of these neurons, we also observe the lateromedial distribution in which the latest-born neurons have their axons more lateral to the early-born ones. So yes, tempo allow us to connect VSX2, in this case, birth date, and their distribution in the zebrafish hindbrain. So these type of zebrafish lines are also very useful and versatile if we wanted to activate tempo in other tissues and organs. So I'm happy to share them with whoever is interested. Also, I wanted to mention that although zebrafish is a really useful model, we can access the zebrafish live. We can do live imaging, while other organisms do not allow this for longer periods of time. So I'm talking about mouse. So in the mouse, to be able to do this type of live reconstruction of the temporal sequence of colors is in many cases not possible. So tempo provides you with a way to reconstruct the lineage in retrospect. So for that, we thought, well, it would be great if we could add that tempo to work in mice. And what we did was to now adapt these constructs. We implemented the constructs with mouse-specific promoters for tempo and Cas9 expression and electroporate them in utero into the brain of mouse embryos that integrate into mouse neural progenitors through the piggyback transposase system. And then we analyze both at prenatal and postnatal stages the brains of these embryos. So as a proof of concept, the vertebrate cortex, the mammalian cortex, is a very good example of birth date related to distribution of cells along development. Why is that? Because in the vertebrate cortex, the progenitors of the neurons that emerge early during development, they position in the lower layers of the cortex, while those that are born later, they go and form the upper layers of the cortex in this inside-out pattern. So we expected the electroporation of tempo would provide the firstborn CFP positive neurons will form more the lowest layers, while the ones that activate RFP and YFP in a sequence would occupy subsequently upper layers, or will form glia, because glia, the astrocytes, form after neurogenesis is complete. So this is what we saw. 24 hours after the electroporation of the embryos, we see CFP cells in the ventricular zone, the progenitors of the brain, and in the subventricular zone of the mouse brains. Subsequently, we see at 14.5 days the appearance of RFP cells, mostly, that start to also migrate out of the ventricular zone. And the YFP, the last reporter, starts to be mostly shown at 15.5 days, and mainly around the ventricular zone and the intermediate zone, while the RFP cells have already migrated together with the CFP cells to the cortical plate. Then they continue the migration, and at 17.5 days, we can see that the majority of CFP cells position in more lower layers, while the RFP ones have passed those early-born neurons. And finally, the YFP cells occupy the most upper layers of the cortex, or reside still near the ventricular zone. So this is consistent with tempo working in a predefined order. In mouse, this is the quantification, and it is also quite efficient. We corroborate that this pattern is conserved at postnatal stages. In postnatal day 10 mouse brain sections, we see that CFP positive neurons occupy mostly the lower layers of the cortex, while RFP and YFP neurons occupy the upper layers. And the glia is mainly formed by the latest colors of the cascade, RFP and YFP. Another important application of tempo is to actually activate the cascade in other temporal populations. So if we electroporate these constructs later in development, at 14 or 16 days of development, we actually shift the cascade start to later points. And in this case, we can explore the formation of later neurons and glia. And interestingly, at 16.5, we found that the glia that forms the latest, the one labeled by YFP colors, occupy mostly superficial regions of the cortex. And while in general, glia has been shown to be quite plastic and not occupy specific layers along development, we find that this is pretty consistent, and we find that most of the latest cells occupy those regions and not others. So it could be that there's a genetic regulatory cascade that activates specific factors, guiding the formation of specific glial types at the end of gliogenesis, and make them form the glia in that superficial layer. So this is important because in development, genetic cascades are actually something that happen in almost every tissue. So in order to be able to specify a cell type, we need a cascade of regulatory or transcription factors. And here I'm showing you the example of the endocrine cell lineage in the pancreas. So in order to specify the different types of the cells in the islet of the pancreas, you need a specific cascade in a particular order. It is also the case for other tissues and organs, such as the central nervous system in the mouse. It has been recently shown that there's a conserved genetic cascade that allow the production of early, mid, and late neurons at every level of the central nervous system. So here the importance of the tools we are developing is that we could adapt them also to explore the genetic cascades occurring during development and disease, and be able to manipulate them to study their function. So this is what we have been implementing in the lab now in collaboration with Daniel Feliciano as well. We have introduced perturbations in different temporal windows of the cascade. How is that possible? Well, we just place a gene of interest, in this case for overexpression studies, in the same frame of the color and temporal window of our interest. This one was for the first window, and this one is when we want to activate it in the second window. And in terms of the factors that we have been studying and using for proof of concept as well for this new development of tempo, we focus on cell cycle regulators because they have been described to be specifically enriched in certain populations of neuronal progenitors, actually. So there are certain cyclins, the cyclin B1, that is involved in the transition from M phase to G1, that has been shown to be enriched in the upper layer neurons and in glia, while cyclin D1, that is involved in the transition from G1 to S, has been mostly seen enriched in those early neurons forming the lower layers. So we thought that it would be very interesting to upregulate these factors in specific temporal windows and see their effects on corticogenesis. So for that, here you have the example of overexpressing cyclin D1, either in the first temporal window or the second temporal window of the cascade. And what we found was that overexpression in each of the windows caused a shift of the neurons to form early born neurons that occupy the lower layers. And this was correlated with their expression in a particular temporal window. So when we deploy cyclin D1, for instance, in the second temporal window, we saw the phenotype exclusively from that time point on. Also had some effects on the cells that were produced after that are labeled yellow. And in the case of upregulating cyclin B1 at different temporal frame times, we found that in this case, it caused the opposite. It caused a fate switch from neurons to glia, so that the first temporal window expressing cyclin B1 caused the shift from CFP neurons to glia, but doesn't affect the next temporal windows, while expressing cyclin B1 in the second temporal window makes the change of fate from neurons, red neurons, to glia. And this cartoon represents the summary of the results we found. Basically, when we overexpress cyclin D1 at particular windows, it shifts the fate towards earlier fates, towards lower layer neurons, here in orange. While when we overexpress cyclin B1, we shift the fate of these particular temporal windows to later fates, to glia. So we found that, tempo has multiple applications. As you can imagine, we can not only label and manipulate specific cell lineages to study organogenesis, but we can also establish the order of cell intrinsic factors required for cell type specification by expressing constitutively active, dominant negative proteins, or RNAs. And we could also, of course, genetically activate or inactivate temporally distinct cells for functional analysis. So overall, tempo allows manipulation of cells in space and time. And to finalize, I can tell you that we can still improve things. We can still increase the clonal resolution that tempo still doesn't provide. So tempo allows us to see the consecutive generations of cells. But if there are many lineages that happen in parallel, many progenitors that transition at the same time, we cannot distinguish them. So for that, there's a tool called Rainbow that I bet most of you know, that allows the activation of multiple color hues at the same time, so that we can distinguish many progenitors that are present at the same time in different places. But Rainbow doesn't have temporal resolution. You cannot distinguish cells born at different times. So the latest versions of our tools, TempoBow, is the combination of both tools. It first activates a recombinase that allow us to have all these color hues to distinguish the different progenitors. And then we subsequently activate by these CASA switches a flipase that was first disrupted, and then we activated in the same way we activated the colors before in tempo. And this flipase, in this case, what does is to activate color hues, in this case, nuclear, in the nucleus of the next generation of cells. Finally, The last step of the cascade is the activation of a halo tag that is represented here by an asterisk. So just to show you some of the results that we recently obtained for the mouse cortex at postnatal day 10, we found that there are cells that were born earlier, occupying the lowest layers of the cortex that only have cytoplasmic hue labeling. The ones that were born later that occupy portions of more the upper cortex contain nuclear labeling, and subsequently the ones that were born the latest have also the other color. So they have both cytoplasmic, nuclear labeling, and halo tag labeling. We are now combining these two with in situ transcriptomic approaches so that we can gain not only lineage and temporal and morphological information, but also cell identity information. So we think this is going to be the genetic tool that gives us more information. So with this, I finish the presentation. Please, well, I would like to thank my lab for all the support during pre-COVID and COVID times. And of course, the Janelia support teams that without them, this wouldn't have been possible. The Koyama lab, the Puyades lab, and all my mentors. Thank you very much.
Thank you very much, Isabel. That was an incredible, insightful talk. I like that's just insane what one can do nowadays, also with genetics, to my mind, because tracking these things, because it's also so super crowded. It's just, yeah, it's pretty impressive. So maybe I have a few questions before maybe the rest of our audience recollects their questions as well. So for me, it's so like, as we talked about earlier, so it takes approximately maybe 1 up to 3 maybe cell cycles until you change from one color to another, right? So that actually means for even fast developments where you have let's say in the early stage of embryo development, you actually have a fast growing organism. So meaning at a certain time after 15 cell cycles or so, you might actually be at the edge of what's possible. My question is, how often can you stack these color sequences on top of each other? So what's the limit? That sounds a bit weird, but would it be possible to have like 3 colors and then another 3 colors and so on and so forth? And but maybe it's also coming back. There's another question which I would have when you presented the images. So these were images which were extracted from the brain. So it were kind of like snapshots from how the brain looked like at a specific moment, right? So you don't really follow these, at least in the brain slices, over time. But you might have the chance to do this with an embryonal development, what you had at the second slide or so when you talked about Kate's work, where you actually would be able to follow the entire development. So because I think with the second part with the first part I was mentioning about if it's just a cut there, I guess stacking wouldn't be useful because you wouldn't be able to tell if you use the same colors from which sequence it comes from. But when you follow something over in development, you actually would see a cycling of the different colors. So you have, yeah, okay, I just let you talk. Lots of questions, yeah.
Yeah, no, but you got the point. So basically, with zebrafish, we have been able to follow it and then estimate to a certain extent the cell divisions. It definitely is going to depend on the lineage. It's not going to be the same for cells that have a high division rate over long periods of times compared to cells such as neurons and neuronal progenitors that have a life, much shorter life. But as you say, we could continue to add colors. So here the limit will be the colors actually, because we could just add another construct in which we add more CASA switches and gRNA switches. Of course, another limitation, of course, is the switch, the gRNA, that is 70% efficient. So as you add more switches, the total efficiency of the cascade is going to decrease. But that is something we have shown that can be improved with more technical research. So you could definitely stack more colors. This would be very interesting for long lineages. And for the mouse, indeed, it will be useful to be able to track this as with Kate's video microscopy techniques to extract cell division information, or even in live brain slices. That is something we have in the pipeline. We want to do that, to be able to really relate the number of cell divisions of neuronal progenitors with the transition. Yeah.
Yeah, because even there, you might actually not even need more colors. You might only need, like, if you know, if there's a certain time, everything's kind of replenished with something new, you can start with the old colors again. You only need the time trace, I think. But it's super cool. Thank you very much. Are there more questions in the audience? Please raise your hand or use the chat function. Okay, yeah, Cornelius, please unmute yourself.
Hi, yeah, I'll try. Thanks, Isabel. That was really interesting. I'm trying to follow and think of all the things you can do with it now. Did I get it right? Given that it's approximately between 1 and 3 divisions, so is that right that in the later dividing lineages, you're contaminated with some cells that have not switched yet? And then do you do some modeling to kind of figure out what the estimated, how do you do that? There's a lot of data analysis there you need to kind of estimate, given that the efficiency, the penetrance, let's say, is not 100%.
Yes. So far, we haven't done the modeling yet, but what we have been doing, for instance, is to use another, in the mouse, another transgene that is an episomal plasmid. So this is a plasmid that doesn't get integrated. So this tells us which type of cells cannot divide. So those cells cannot transition. So that's part of the contamination we see. All those CFP cells that we saw in the upper layers were cells that were already transitioning to differentiation. And those were labeled with this other plasmid, the episomal plasmid. So this type of co-labeling experiments allow us to estimate what is the percentage. And we estimate around 30% of cells that are labeled with the first color are not able to transition and they are contaminated. So we are now designing other ways to co-express tempo so that it doesn't stay in cells that do not divide.
So maybe just follow on to I had asked that question before, how much control you have over the frequency of the events. Can you change the sequences around the cut sites and just, empirically get more and less efficient CRISPR activity? Is
that could be an option. I would prefer, honestly, to have a promoter, as I said, like Hitchcock, or that really allows you to express Cas9 or guide RNAs at a certain frequency, more controlled.
Yeah.
But what you said is very interesting, and we have evidence that the different guide RNA sequences or adjacent repeat sequences can make it more or less efficient. Yeah.
Thanks.
Okay, how easy is it to implement this right now? If I have like a crazy mouse system right now, and I really want to have what you have, and I'm a physicist, so how do I go about this? Where would I get the information, or where would I get these sequences, and how do I implement this in my system?
Okay, so this is going to be in bioRxives like in less than a month, so you will have all the information. But you mean implementing it in a line, or what exactly?
Yeah, so when I read your paper, so the bioRxiv paper, and I say like, oh my God, that will be so groundbreaking for my research. How do I get this? So should people will you put this on gene sequences? Like how would I'm just like asking for a general audience who are kind of impressed by this and who are maybe not as deep into the matter as you are. So how would people implement this in their own systems?
Mm-hmm. Yeah, so I'm definitely going to deposit every sequence, zebrafish line, you know, open in, well, through a gene also, they will be available. And so options are zebrafish that I already have. I'm happy to share from frozen sperm to adults that could be directly crossed to the lines of interest that people might already have. And then they just use their Galfor system to activate tempo wherever they want. The other option, of course, is to just send the plasmids and people can inject them in zebrafish. And the other option, of course, for mice is also the plasmids. For now, we haven't generated the stable transgenic line yet. We plan to do that, but for now we can electroporate the constructs, and that could be done also in tissue explants if people are interested.
So right now, then the big question: what's then next?
Next, first of all, is to continue developing this tempo bow strategy, which I think is going to solve many questions. But for me, well, I'm still very passionate about the connection between nervous system and organ development. So that's the future of my research. I want to study how this connection forms, specifically brain pancreas in my case. And for that, now with these tools, I can really trace the cell specification cascade, both at the level of the endocrine precursors and the neuronal precursors that are in tight communication with those.
I guess that's also in line with the question which Cornelius right now posted: what will be your favorite application for this tool to address the burning biological questions? So I think you already gave the answer to that, unless you want to be a bit more specific about this. But if there are not more questions, so are there more questions in the audience? Otherwise, I would actually hand over the word to Christian right now to give us just a very brief overlook of what we can expect in October.
Yes, will be very brief. Thanks. So I think it's 5th of October, right? Yes, cool. Yeah, so basically the next seminar, so it will be a speaker from EMBL again, a postdoc of Anna Kreszuk, who some of you might know. So she's a machine learning expert. His name is Adrian Wolny, and he will present new developments for segmentation in images, which should require less labeling efforts. So I think it will be very interesting. Yeah, that's it from my side.
Okay, so thank you very much, Christian, for the update. So with that, I think I can just speak in behalf of the whole crowd to thank Isabel again for the amazing presentation. So thank you very much for your time. That was a very insightful presentation, and yeah, see you all then during the next seminar in October. And yeah. Bye-bye. And for those who want to stay for the Meet the Speaker session, please do so. I will definitely stick in this channel right now for the next 5 minutes, for a bit longer. And everyone else who has still in-depth question, they can ask their questions. Otherwise, to the rest, bye-bye.